Now let's have a look on some morphological features of cardiac muscle. Cardiac muscle is organized in cardiac muscle cells, also called cardiac myocytes, that have a tendency to be slightly branched, like that, for example, and they are connected in a three-dimensional network Each cardiac muscle cell has usually one nucleus that is in the center. Exceptionally, there might be two nuclei, but usually there's one nucleus in the center. On the poles of that nucleus, you can find an endogenous pigment, especially in elderly persons. It's called lipofuscin. There's this striation pattern similar to the skeletal muscle. There is again the basal lamina surrounding each cardiac myocyte, and there is connective tissue with a very rich capillary network. Which is the connective tissue is called endomysium, and here we have got these capillaries, very rich network. On the contact between two adjacent cardiac myocytes, there are special regions called intercalated discs that are accumulations of cell-to-cell contacts. So we got the cardiac muscle cells, surrounded by the basal lamina. The nucleus is in the center. Therefore, if you got cross-section, you would see the nucleus like this, approximately. There is this striation pattern. Intercalated discs. The endomysium connective tissue, it's a reticular connective tissue with many capillaries. The dense capillary bed. These are capillaries. Okay, and this pigment is called lipofuscin. It accumulates during aging. It's also called wear and tear pigment. It's a product of an imperfect recyclation of cell organelles. If we look more closely to the intercalated discs, where two cardiac myocytes meet, it's not straight, it looks like this. And there are cell-to-cell junctions that provide both mechanical as well as electric coupling. So here we have the basal lamina. And one of these connections is called a fascia adherens with cytoskeleton inserting with the help of some transmembrane proteins and integrins into a plate in the extracellular space. Similar are desmosomes. Fascia adherens. This is the macula adherens. So these two junctions are responsible for mechanical cohesion between the cells, because this is myocyte 1, okay, and this will be myocyte 2. And the third junction is the gap junction, which consists of subunits 1, 2, 3, 4, 5, 6. They are made of protein called connexins, and they can close or open. So it's gap junction. Another word is nexus for that cell junction, and it permits small molecules and including ions to pass through. So the sarcoplasms of two cardiac myocytes are electrically coupled. If you look in this subendocardial region of heart ventricles, you'd find specialized cardiac muscle cells called Purkinje fibers. In comparison with the working myocardium, with the working cardiac myocytes, with the regular, the Purkinje fibers are larger and less intensely stained. They also have nuclei in the center, but the working myocardium has better visible cross-striation pattern. It has more myofibrils. So this is the regular, or let's call it working myocardium. And these are Purkinje fibers. Actually, these are not fibers, these are cardiac muscle cells, and they have a large diameter and more pale sarcoplasm, because they have less myofibrils. Their purpose is not to generate force, but to distribute the action potential in the endocardium, in the subendocardial layer of heart ventricles. Because they work as terminal parts of the cardiac conducting system that distributes the action potential throughout the myocardium.